Art Motorzenik, and I'm with the American Association of Pharmaceutical Scientists. It's my privilege to serve as a host for interviews of distinguished pharmaceutical scientists who have made a major contribution to our profession through their research careers. Speaking on behalf of the AAPS Distance Learning Committee, we have invited these scientists to share with us some introspection into their careers, what motivated them, what kind of mentorships they experienced during their life, and how they envision their past careers, their present careers, and an outlook on the future. Today it's a personal pleasure for me to interview Les Benet, a good friend and associate as well, but one of the eminent academic scientists. He's had a very distinguished career at the University of California and San Francisco School of Pharmacy, both as an eminent professor and as a past chairman of the Department of Biopharmaceutics. So Les, I'd like you to begin the interview by telling us a little bit about your formal training and background, what brought you to pharmaceutical sciences as a career.
Okay, thank you, Art. It's nice to be here and to have the opportunity to participate in this. Let me give you a little history. I basically wanted to be a university professor, and I wanted to be an English professor. I wanted to go to Yale. But in the days that was when you had to do the GREs or the SATs, your parents had to sign. Now, my family owned a pharmaceutical company, made hyperallergenic, some of the first hyperallergenic shampoos and stuff like that. And they said, no, you got to go to pharmacy school. So I went to the University of Michigan and started in pharmacy, but decided after about a year and a half that I didn't want to do this, and I switched into English, and I got a degree in English. And I was going to go to graduate school in English. I met my wife at that time when I was an English major. She was an English major. And I had the good fortune of having a professor to tell me that after I'd been accepted in graduate school in a couple of schools, that told me, you know, you'll be a great teacher, but you're never going to be a good creative writer. And that's pretty odd that he could be honest like that. And he's wrong, because all my science is creative writing. And I said thank you, and I went back and I finished my pharmacy degree, and I got a master's in pharmaceutical chemistry at the University of Michigan. Then I went to the University of California and got a PhD in pharmaceutical chemistry at the University of California.
And were you motivated essentially right from the start when you went into pharmaceutical undergraduate work that you're going to go on to a graduate degree?
Yes, yeah. I was always I wanted to be a university professor. That's what I thought would be a neat thing to do. Yeah.
Well, it's certainly a career that you've managed to achieve, even though you spent almost all of your time at one major institution. I know you did spend some time at other academic institutions as well. Who were the kind of teachers that might have inspired you toward the choices that you made in research directions?
Well, it's interesting that I did my first research as an undergraduate with R. Paul in pharmacognosy at University of Michigan. He was an assistant professor, and that was the first research that I ever did. And then I was very influenced by Jerry Goyen, who was in physical pharmacy. I always liked math, and I liked what he was doing, and I became his graduate student. And he had a big influence in terms of the career. I was basically physical chemist, as you were, a physical pharmacy type student who was going to apply physical chemistry principles to pharmaceutical systems.
Right.
Then I had a very sort of interesting thing that happened. Arnold Marcus was chairman of the Academy Arden House program, and they were doing the program on pharmacokinetics. And Arnold asked me if I would speak in the program on pharmacokinetics. I said, Arnold, I don't know anything about pharmacokinetics. He said, well, actually these other guys don't either. And why don't you just on the last day be the reviewer of everything? So I went to Arden House. I took all the journals and everything, read it, and then that's actually how I started to become a pharmacokineticist.
Well, that's almost like by happenstance.
Yeah.
Of course, you certainly, in your career, helped to define the direction, but there was a time in that era when people were still exploring where pharmacokinetics might be applied. Who did you see at that time as people who influenced your thinking?
Well, actually, John Wagner had a big influence on me. John, when he retired from the Upjohn company, wanted me to come to Upjohn and take that job. A lot of other people he did also. But John had some of the first courses in pharmacokinetics that went around the country, and I taught in those courses. I was the young guy. There was a lot of these old guys, and I was the young guy in it. And my ability at that time was I knew a lot of mathematics. And most of the pharmacokinetics people really didn't know a lot of mathematics.
Right. So you turned it quantitative.
That's
very
right.
much so, yes.
Yeah.
Well, and then, of course, pharmacokinetics turned out to be a platform for the emergence of other applied disciplines. Drug delivery, for example, that followed very rapidly, and then product design, and then we get into the whole pharmacodynamic area in which you've pioneered, and you got very eminent credentials. Were there collaborators that you collaborated with, in addition to the people that you had at UCSF, who were very important in your early career?
Well, the most sort of important first paper. Well, I had a couple really important papers on the mathematics of pharmacokinetics because people were doing all kinds of crazy things, and they could be much simpler than what was done. They were publishing lots of papers in JPharmSci that were just sort of mathematical repeating, and I developed a bunch of nomograms or ways to approach pharmacokinetics that really simplified everything. So that's sort of what I did initially. But then working together with Malcolm Rowland, we developed clearance concepts and published in 1973 that first very important paper, Clearance Concepts in Pharmacokinetics, which changed pharmacokinetics from strictly a mathematical description to something that had physiologic and pathologic basis. It was a very important development in our career. Now, the other really sort of influential thing that happened to me, I was on the pharmacology study section at the NIH. I was the second pharmaceutical scientist to be on the study section. Milo Gibaldi was the first. But when I came onto the study section, Alfred G. Gilman, the Nobel Prize winner, came on at the same time. And in those days, you used to room together, two people to a room at the NIH. You didn't have. And Al Gilman and I roomed together for 4 years.
That's significant.
And on the basis of that, the first chapters on pharmacokinetics in Goodman and Gilman appeared, which he had me write because I convinced him pharmacokinetics was important and the pharmacokinetic parameters were important. So that was very influential and sort of historical. His father, Alfred Gilman, did not want pharmacokinetics to come into the book. He thought that that would be, that was not what pharmacology was. But Goodman and Gilman Jr. were very much for it, and it's become a very big part of it. So that was a very influential thing for me, but also influential for the field, because we became recognized as being a critical part of pharmacology.
How long was your activity active with the NIH and the study groups, and how long were you associated with that?
I was 4 years. I can tell you another interesting story. I was the first chairman of an NIH study section that came from a school of pharmacy that wasn't a medicinal chemist. And when I was nominated to be chairman of the pharmacology study section at NIH, the medical school chairs of pharmacology sent a delegation to the NIH and said, Les Bennett's a good guy and we don't mind him, but if he becomes chairman of the pharmacology study section, it's the death knell for traditional pharmacology.
My goodness.
And the NIH told them, go to hell. Well, and I became chairman of that study section.
Well, I think not only there, but in many other areas, you've been a breaker of tradition and well advanced our pharmaceutical science career opportunities. You also were very pivotal in participating in other government work and supporting work at the FDA. Would you want to comment about that as well?
Well, it was later in my career, but I've always been very involved with the regulations and felt that it was something that pharmaceutical scientists should be involved with, that there should be a good scientific basis. In fact, when the first regulations on bioequivalence were published in 1973, I went to the agency and said, this is, you're making a mistake. You're defining things wrong. And they sort of hemmed and hawed, but I was pretty young then, and they decided it wasn't an issue. And what happened was they were getting themselves in a box because they were saying you define bioavailability in terms of sites of action and you do it with absorption and things like that. I mean, we've gotten around it, but it's in the law, and it would have been a lot easier to scientifically define it correctly initially.
When I asked you earlier about some of the mentors you had, and you acknowledged several, what I observed in the evolution of pharmacokinetics was that there were certainly some of the traditionalists that helped pave the way in the profession. but that very rapidly you emerged, as you pointed out, the quantitative and the introduction of clearance kinetics and so forth. You emerged as a very strong voice very early with some other collaborators at that time. Would you want to comment on that, sort of the new wave as well as the traditionalists?
Well, we believe that you should run studies in vivo and relate it to physiology and pathology. And that I have always run many human studies in patients as well as in healthy volunteers. And I felt strongly that you needed to have a kinetic basis as well as a pharmacodynamic basis. I did interact a lot with Gerhard Levy in those days because he was really the leader in pharmacodynamics. Ed Garrett, who was a curmudgeon of the first order, yes. Ed used to think that Ed liked me a lot and used to sort of say that the kinds of stuff I was doing was good. So I really had a good interaction. I knew, I am the historical link because I have known everybody in pharmacokinetics personally except Meyer and Overton, who published a paper in 1917.
Yeah, Meyer-Overton theory, right.
Yeah, but starting with Tyrrell in 1937, who's often considered the father of pharmacokinetics. I knew him at the University of Uppsala. I knew the German who wrote Der Grundlagen to Pharmakokinetik. I knew all of those guys at one time, and I know all the young guys today.
Right.
So when I die, we're going to lose that connection.
So you have an interesting perspective, especially in terms of how evolutionary applications have come out of pharmacokinetics. Now, one of the areas that you've contributed greatly to is the application of pharmacokinetics to drug product design and drug delivery as well. I'd like you to comment on that because that was an important industrial application immediately of clinical pharmacokinetics, trying to achieve dose-sparing concepts, for example, and looking at the value that would have in terms of patient acceptance.
Well, it's interesting because today, I mean, I have all these new concepts that I've sort of developed in terms of how applying BCS and make predictions. And I think back and think, gosh, how couldn't I why didn't I think about that 20 years ago? You know, it's so obvious of some of the things that were happening. But we definitely understood how you translate the difference between what was happening in the liver and what was happening in the gut. And that was probably, people think that that's probably my major contribution, of being able to understand that the gut really was a disposition organ. In other words, drug was eliminated in the gut as well as being an absorption organ. Right. And that if you didn't pay attention to enzymes and transporters, you were going to get the wrong answer. And that you had to pay attention to both of those. And a lot of our concepts of drug absorption and what was going on was more from a physicochemical basis, and I think I brought to it really more of a physiologic, pharmacologic basis.
Well, that was a good application of where you did a lot of outreaching. Your work in metabolic enzymes and transport proteins actually require you to reach over into physiology and looking into a significant amount of clinical science dealing with pharmacodynamics. How challenging was it at that time? Did you feel that there were you commented about some of the resistance from traditional disciplines there, but where did you see opportunities that were especially fruitful for you?
Well, it was good to be at UCSF because UCSF is a very collaborative place, and I had no difficulty. I remember in some of my early days doing monkey studies for drug absorption, and I got the chairman of GI surgery to do the surgery on my monkeys. I said, here's the study I want to do. I want to put a Thomas cannula in these monkeys. And he thought that was a good idea, and he did it for me. And I was able to interact with the transplant surgeons because they felt pharmacokinetics was really important, and they wanted to have the information. So it was really a good environment. I had all these clinicians who believed that pharmacokinetics, and this sort of goes back to Goodman and Gilman, pharmacokinetics being in there. They start paying attention to it. They need to pay attention to drug dosing and things like that. So it was a unique opportunity at UCSF because I always had good physician collaborators who would allow me to study. And even today, I always have I'm a member of the clinical pharmacology training program. I always have MD fellows in my laboratory. I have clinical faculty who are on my grants, so I have good access to be able to do that. And I've been very lucky. That was a very fortunate situation. I mean, the guys at Buffalo didn't have that. I mean, they made a lot of contributions, but they didn't have that interaction.
Well, to be fair, you commented about how you got an enormous amount of support from people at the university, but you have a pretty recognizable reputation of being a tremendous supporter of people who come to you for help. So I would imagine that a lot of the, perhaps the reward and satisfaction you've had in your career has been from helping other people by bringing their disciplinary interest and sharing some of your outlook with them.
I think that's very true. I remember in my early days at UCS, we did a lot of work with radiologists. We worked with hepatologists, and we worked with people who were interested in biliary canaliculi stasis, and they would come to us and say, you know, we want to measure this, or we're interested in this, and we say, yeah, we can do that. And it'd be great opportunities. Just was just a great environment to be able to do the kinds of things that I was able to do.
Well, Les, as you were evolving a significant reputation at UCSF, both as an instructor, professor, a researcher, and a collaborator, you also played a major role in globalization of some of the disciplines you've approached, pharmacokinetics and drug delivery. Could you comment about how you benefited and maybe how you gained some insight from international collaborations that you might have had in your career?
Well, I had a lot of international collaborations. One of the most, probably an essential thing that I should mention is when the Japanese-American meeting was to be held in 1986.
Jack Farm-Sci.
Jack Farm-Sci. I was the program chairman for the U.S., and TAC made me the program chairman. Takaguchi made me the program chairman, and Professor Hanano from Japan was the program chairman for Japan. So I had an opportunity, many times TAC and I together would fly to Japan. So I had a lot of opportunity to interact with TAC in those days, and a tremendous opportunity to influence and interact with the Japanese.
Right.
And that has been something I've been very proud of. I've been a big supporter of the Japanese. I've probably had close to 25 Japanese postdocs in my laboratory over the years. I interact strongly with them and brought them into sort of the modern pharmaceutical world in terms of pharmaceutical sciences to interact with us. And I'm very proud of that.
You're a very well-published scientist. I'd like to put you on the spot and ask you, in a cluster of research areas, what areas are you most proud of in terms of being pivotal or having somewhat of a seminal influence in the disciplines?
Well, I can tell you what my most cited papers are. Okay, that's probably a good way to do it. And I am one of the 250 most cited pharmacologists in the world, so I had some influence that way. My most cited paper is the paper when we did moment analysis in pharmacokinetics. And we were able to calculate pharmacokinetic parameters, clearance, volume of distribution, and mean residence time without using compartmental models. So it was sort of getting away from that mathematical definition.
And you said we, who would?
Renato Gagliazzi was the co-author. He was a physician. Was a co-author. He now lives in Germany and in Switzerland, I'm sorry, and he was the co-author. And that paper has been very well cited, about 1,000 times.
Right.
The second most important paper I had is more recent, is 1995. Oh no, second most important paper was my paper related to predicting that the allergic toxicology of nonsteroidal anti-inflammatories, aspirin, all of the nonsteroidals, came from covalent binding through a glucuronide metabolite. And that glucuronide metabolite could then covalent bind, make a hapten, and have an immunologic response. Still today, I go to companies to consult about that, because if they got a carboxylic acid drug, it makes a glucuronide, it makes covalent binding, and the agency always asks them what happened. So we defined that, and that was pretty early, and that paper gets cited a lot. And the third most important paper is one more recent, 1995, and it's really the basis of what I do now. And that was a paper we published in Molecular Carcinogenesis that said, If you look at drugs that are substrates, an inhibitor of cytochrome P453A, they're also substrates and inhibitors of P-glycoprotein. And we predicted in that paper that there was coordinated regulation between enzymes and transporters. And it was prior to PXR and CAR and all that kind of stuff, and we've really been proved right. You know, we jumped in and we sort of saw this. Chi-Wan Wu and Vince Wacher were the co-authors on that paper, and that has been the basis for major change in our understanding of drug metabolism. The transporters have an important effect on drug metabolism. So those are probably my three most important papers, and the next one is a paper related to liver transplant with cyclosporin. Yeah.
Very good.
Yeah.
In addition to writing your own publications, you've made a major contribution to the pharmaceutical sciences in the promulgation of good journals. You've been an editor of several significant journals. You've offered insight in improving the quality of pharmaceutical research journals and everything else. Tell us a little bit about what opportunities you saw, how you seized them, and how you see maybe areas of improvement even in that area in the future.
Well, I was a co-founder with Sid Riegelman and Malcolm Rowland of the Journal of Pharmacokinetics and Biopharmaceutics in 1973. And that was the first pharmacokinetic journal. And when Sid died, shortly after, I became the editor along with Malcolm, and then editor emeritus in '98 of the journal. And I've been a co-editor of Pharmacology and Therapeutics, and I've been on an editorial board of a number of the pharmacology journals. In fact, I'm on the editorial board of a number of pharmacology journals, which I think is really good, because I think it's important that a pharmaceutical scientist be on those editorial boards. I think what we're seeing in science in general is a diffusion away from strict disciplines to interactive work. And working on journals and interacting in that way, I think, has been very helpful. And I've enjoyed it. I try to stop doing it now because it takes a lot of time. But it's something that is really important. I was a big supporter of the formation of pharmaceutical research. When we founded AAPS, I had the journal ready to, with my colleague Wolfgang Sade, because I thought we needed a first-class journal, and so I've tried to do that.
And you're well recognized as being a pivotal force in the high quality of journals. That's why I wanted to address that issue. Which brings me to AAPS. Of course, we both have had a close relationship in the dawning of that organization. But you emerged even before AAPS as a significant leader and a voice for bringing pharmaceutical sciences together, multidisciplined, international, as we spoke of. How do you retrospectively, Les, look back at the issues that brought you to such a key role? I mean, you put so much energy into trying to establish a good, sound organization for pharmaceutical scientists.
Well, I really felt it was important that pharmaceutical scientists have an identity. And to have an identity, you had to have some way that you were recognized and independent. If you were subservient to another profession, you were never going to achieve that. And you and I shared this vision that we knew that there were 15,000 people out there that were potentially members of a new association. They're up to 13,000 now, but they're going to reach 15,000 and more. And it just needed to have its independence. It needed to be able to make the kinds of statements that reflect pharmaceutical sciences, not necessarily the pharmacy profession, because pharmaceutical sciences goes across all medical disciplines. It's a service of all medical disciplines. It can help all medical disciplines. And if we were tied only to the profession of pharmacy, we weren't going to be able to do that. So in my mind, it was really critical to have pharmaceutical sciences as an independent group and to be able to succeed and to show what we could do.
What's very remarkable to me are the years 1984 to 88, when your record demonstrates significant achievement in research accomplishments. and where you were honored with significant memberships in scientific societies and so forth, and well-deserved. And yet those were the years where seemingly people would feel you were totally distracted with the founding of the AAPS and some of the pulling together of the talents and the team that you put together. How do you look at it retrospectively?
Well, that's interesting because I'm probably a good organizer, and I had a wonderful staff. I was chairman of the department at UCSF, and again, UCSF is quite different than most pharmacy schools. That the power is in the chairmanships. And you have your own budget, and you create your own budget. So I had a terrific staff that could run the department and run things within the things within the organization. I had great vice chairs within the department, and I had good opportunities to have wonderful postdocs and graduate students. I mean, I know that if I get into the lab, the experiment gets screwed up. So what I do is talk to the graduate students and postdocs and say, hey, gee, maybe why don't we try this and stuff like this, and then they go out and figure out how to do it, and then they send me an email, and those days they would have to send me a fax and stuff. But if I was there, you know, the work would get slowed down. So I was very fortunate to have really good people, and I didn't sleep in those days. I don't sleep today.
I could imagine that was true because, as I recall, I mean, at conferences like the Ozark meetings and so forth, here you were involved so heavily in the leadership role you played at AAPS, and yet you were a prolific contributor, seminars, symposia, articles, poster sessions. Your students were active. It would seem that you were putting something like 26, 27 hours into every day. I'd like to ask you where you found the level of energy and support, maybe from family or others in addition to your scientific collaborators.
Well, I had a very supportive family. I had my wife, Carol, who got her PhD, and when we raised the kids and they went to high school, then she went back to graduate school at Berkeley and got her PhD in comparative literature. My daughter and son were good interactions. We didn't say that when they were young. I taught at Washington State University for 4 years, and I had a lot of opportunity just to go back and forth. And even when I taught at California, I would be home every night. I would be home every night when the dinner time and they go to bed. I would then go back into the office. So I was somebody that would go back in at 10 o'clock or something like that, work. So I tried to be a good father and to interact, and they turned out to be good kids. So either I'm very lucky or I've done something right. But it was a very supportive family, and a family that liked to do stuff together.
Well, it's been a partnership with Carol, and I'm sure your children and all have contributed much to helping you get motivated for some of the significant challenges you undertook. I'd like to speak to some interesting things you've developed in the entrepreneurial area recently. You're a founder of AVMAX and of Oxon Medica. Could you tell us a little bit about those adventures?
Okay. Well, AVMAX came about from the discovery of the enzyme transporter interaction in the gut. We carried out these studies in humans where we were looking, what we tried to do was see if we could change the liver metabolism by inducing it or inhibiting it for the immunosuppressives that were important in cyclosporin, tacrolimus, sirolimus. We did all of them. And we found we couldn't explain any of the data. None of the data fit to the present pharmacokinetic hypothesis that we had. So the only way we could explain it when we did some analysis was say the gut is so much more important than we thought before. And that for cyclosporin, in fact, the old Sandimmune formulation, our data said that half of the dose was metabolized as it went through the gut. And that it wasn't an absorption problem, it was a gut metabolism problem. Well, when we realized that, we said, well, we could inhibit this. If we could inhibit this, then we could change the bioavailability and increase the how much drug got in and decrease variability. So we wrote some patents on those in the early 1990s. And those turned out to be good patents. And that was, a company was founded. UCSF is very supportive of faculty members being entrepreneurial. We're a very entrepreneurial campus, and support back and forth. And so they would license the patents. And I've had some good interaction. I've had some tough interactions too. We were a big partner with Elan, and then when Elan broke, that caused us a lot of problems. But we've had some good things happen to us more recently, and The other thing was Oxone Medica. Oxone Medica, again, related to research in my lab. I did a lot of work on nitrates. We were the first people to measure nitrates, nitroglycerin in the blood from a sublingual dose and to show its bioavailability. And there was a nitrate tolerance problem that everybody said you develop tolerance. And there was a whole bunch of hypotheses. And we came up with our own hypothesis that said actually nitrate tolerance was oxidation of guanylate cyclase. What do? We deal with enzymes and transporters. So this was an enzyme problem. We tried to publish that in all the cardiology journals, got all turned down. We didn't know anything. Right? So we said, well, did finally publish it in Journal of Pharmacology and Experimental Therapeutics. Our idea was that if we were right, if our hypothesis was correct, then we knew how to make molecules that wouldn't have tolerance. That we could put anti-reactive oxygen species in the same molecule with NO donors. And that's what Oxone Medica does. And it writes its patent on composition of matter. But it all comes out of doing research and asking the kinds of questions that you try to see. And
well, as you progressed in your own individual career, would you be able to capture what was going on in our profession at that time? Would you consider any defining moments in the profession during the span of your career which influenced your own choices and made you perhaps transition from one field to another?
Yes, for sure. You can't be at California with clinical pharmacy and not be interested in the clinical application of what you're doing. If you're going to teach the students something relevant that is directly applicable, then you've got to be doing clinical pharmacokinetics. And so as the profession moved toward what we then called clinical pharmacy, but more practice-based pharmacy related to the patient, that had a big influence on the types of things that I did. And I would teach both the pharmacy students and the medical students the basic pharmacokinetics and the theoretical application related to physiology and pharmacology, and be able to translate that over. So I think the change of the profession in terms of its movement toward being a patient-oriented reflected my own research in moving in that direction. Yeah.
If you were to, and you are a visionary, so if we were to ask you for your vision right now of what are some of the biggest challenges facing pharmaceutical research today, in our era right now, and maybe I'm going to follow up by asking you to anticipate what they would be in the short term or near future as well.
Okay. I think it is really important to be realistic. Individualized medicine is a very interesting hot concept with genetics and all that's translated. But it's not really reflective of something that's going to happen, because it assumes that genetics is the major source of variability. And it's not. Genetics is not the major source of variability. And even when you get a person that has a genetic variable, if you've got a narrow therapeutic index drug, you've still got to monitor that patient. All the genetics will tell you what's the right first dose. So I think we're overplaying that. I think we are going to make a lot of drugs that have used that basis and saying, okay, we're going to dose those patients. But I really don't see us making this jump in terms of being able to tailor the dose to the individual person much better than we're doing at the present time. So I think that's something. I think the other major thing that's happening in the pharmaceutical industry is a recognition that combinatorial chemistry and high throughput screening combined to develop drugs didn't work. And we didn't get all the great drugs that we thought we were going to have. And actually it requires thinking. That somebody's got to go back and think about that stuff, and you're not going to just mechanically run out and have lots of drugs.
So you're referring to the assessment of what we call in silico chemistry.
Right.
Yeah.
But in silico chemistry will work as long as we understand all the basic science. Now, what I've been doing in the last few years is saying that the basic science is deficient. The basic science is deficient because we haven't understand the importance of transporters. And until we understand the importance of transporters, in silico is not going to work.
Right.
So you got to always be careful that you really understand the basic science. If you just jump into things and think you're going to be able to do it based on the data that you have and doing microarrays or arrays and fitting all the data, I don't think that's going to come out. I think one of our biggest challenges right now is going back and saying, what's the rational approach? Microdosing, big hot issue right now, not going to get us anything. I don't think it's going to do anything because drugs don't work the same way at very low doses as they do at the normal doses that you give them in humans. You see all kinds of different activities. So I don't think it's going to have that kind of imprimatur that we expect. So there's always hot areas. There's always hot areas of thinking. And I think it's important to be skeptical. I try to teach clinicians to suspend disbelief. I teach scientists to suspend belief. And the reason I want clinicians to suspend disbelief is what happens is they see a result and it doesn't fit what they're taught. And therefore they say, oh, it's an anomaly. And it's not. It's the clue. It's the clue that leads you to something else. It's why I got into this whole field. Why did transplant patients with tuberculosis, cyclosporin didn't work in them? Why was it? Well, they had tuberculosis and they happened to be taking rifampin, and rifampin was upregulating their enzymes and transporters, and the dose of cyclosporin wasn't going to work. So don't just look at it and say, you know, oh, I don't understand it, go on to the next patient. Suspend disbelief.
Total picture is necessary.
Right. Yeah.
Well, these have been very good introspections. I'd like to ask you a question about what kind of advice you would offer to young scientists entering the pharmaceutical research profession today.
Well, I still think it's the golden age. It is the golden age of our field, certainly in almost all of our fields represented in AAPS. Drug metabolism, pharmacokinetics, we're having a major impact. We have become, in many cases, the rate-limiting step in drug development, and drug delivery is the rate-limiting step in drug development. So you can find the molecules and you can find how they react, but you got to be able to put them into humans, get them to the site, and to be able to understand the kinetics. So it's a great time to be a pharmaceutical scientist. Wow. And I think it's really, you know, the students that come out of my laboratory, they're doing very well.
Well, you're ending the interview on a wonderful note of optimism, and your energy and enthusiasm is very evident. So it's been a pleasure interviewing you, Les Benet, my dear friend, and I certainly wish you continued success in your existing career, and it's a pleasure having this interview.
Thank you, Art.